Okay, so I'm going to talk about milk proteins, and these are going to be mainly the milk proteins that are found in cow's milk, since it is the most abundant and the most widely used form of milk. Proteins make up about 3.2% of the total composition of cow's milk. Of this 3.2%, caseins make up approximately 80%, and whey proteins make up the other 20%. There are 4 types of caseins, and these are the alpha S1 casein, the alpha S2 casein, beta casein, and kappa casein. There are also 4 proteins that make up the whey proteins. These are the beta lactoglobulin, the alpha lactalbumin, immunoglobulins, and serum albumin. First, I'm going to talk about the caseins found in cow's milk. Casein is a heterogeneous group of phosphoproteins. It has no disulfide bonds, which means that it does not coagulate during cooking. It precipitates at pH 4.6 at 20 degrees Celsius, and native casein contains relatively high concentrations of calcium and phosphate. Acid removes calcium and phosphate, and at pH about 4.6, casein is completely free of salts, which is denaturization. And also, rennet causes limited proteolysis, which also causes denaturization. There is also the casein micelle. Casein micelles are approximately 30 to 300 nanometers in diameter. It is a colloidal particle, and it is the quaternary structure of caseins. So you can think of the casein micelle as just a large group of casein molecules compacted in one area. There are 3 parts of the casein micelle. The submicelle, which is the small aggregates of 100 to 10 casein molecules. This is about 94% protein, and then 6% small ions such as calcium, magnesium, phosphate, citrate, and enzymes. There's also a hydrophobic core, which is the casein molecules themselves, and there's also a hydrophilic coat, which is polar moieties of kappa casein around the outside of the structure. So it's just a bundle of them together with kappa casein around the outside and then casein molecules in the center. There's also a phosphate group that contributes to the structure and stability of the micelle and its water binding capacity. Formed by cross-linking of phosphate groups by calcium, and the micelles are negatively charged, which causes repulsion, which is why it is able to remain as a stable colloidal dispersion. Next, we're going to get into the whey proteins. The first one is the beta lactoglobulin. This is the most abundant whey protein. It is made up of 8 strands of antiparallel beta sheets. It also has the S protein, which is what causes the cooked flavor. It denatures at about 67 degrees Celsius and a pH of 6.7. The reason it's thought for association is its disulfide bridges, and its biological functions are to bind small hydrophobic molecules such as retinol, alkenes, and phospholipids. The next is the alpha lactalbumin. It is a globular protein that is made up of 4 alpha helices, and 1 antiparallel beta sheet. It binds calcium, magnesium, and zinc. The calcium helps with the protein folding, and alpha lactalbumin also denatures at 63 degrees Celsius or a pH of less than 4. Its biological functions are the synthesis of lactose molecules.